Welcome to Le Podcast de Philippe. We're going deep today into AI and how it could change how we create genomes. We'll be talking about Evo2. It's a powerful AI model.
Tuned on a lot of data.
Yeah, a massive amount. Over 128,000 genomes.
Wow.
It's incredible. And that includes human and animal, plant.
Even microorganisms.
Even microorganisms, yeah.
Yeah.
So we'll look at what Evo2 can do, like how it can spot variations linked to diseases and even write entire chromosome code.
That's right.
Speaking of reaching new heights, we're hoping to hit 1,000 subscribers on YouTube today.
Awesome goal.
If you like what you hear, hitting that subscribe button would really help us out. Now let's get into the potential and the challenges of AI-driven genome creation.
So Evo2, it's a groundbreaking model. It came out of a collaboration between
A few different groups, right?
the Arc Institute, Stanford University, and NVIDIA. And what really makes it different is that it can go beyond just analyzing DNA. It can actually create completely new synthetic genomes.
So it's like having a digital geneticist able to design, even rewrite the code of life. And all thanks to this massive data set it's trained on. Like you said, the largest ever used for a biological AI.
Right.
What exactly is in that data?
So it's made up of 128,000 genomes. It's a huge variety of organisms, from bacteria to humans. This gives Evo2 a broad perspective, allowing it to see those subtle patterns and connections that you might miss if you were just focused on a smaller set of data. And this is really important when you're studying non-coding DNA, which actually makes up most of our genome.
Most of it, huh?
And it plays a big role in regulating gene expression.
Okay, so Evo2 analyzes this massive amount of genetic data and finds those hidden connections in the non-coding DNA. Then what? What does it do with all that?
So one of the things that Evo2 is really good at is disease detection. By looking at DNA sequences, it can pick out variations, especially in those non-coding regions that might be linked to diseases.
So it's like a detective, finding those clues to understand diseases better and maybe even come up with more effective treatments. But it sounds like Evo2 goes even further, right?
It does.
It's not just about analysis, it can create too. What can you tell us about that?
Evo2 doesn't just analyze, it writes entire chromosome codes. It's creating new genetic sequences. This changes everything for biodesign. We're not just designing proteins anymore, we're designing entire genomes.
Wow. Can you give us some specific examples of what Evo2 has actually created?
Well, researchers have used it to generate genomes inspired by all kinds of organisms, bacteria like Mycoplasma genitalium, human mitochondria, even a yeast chromosome. This shows you how versatile it is and the potential to really tailor genomes for specific purposes.
This is where it gets really exciting and maybe a bit unnerving too. We're talking about designing life itself. What kind of impact could this have on the future of medicine?
I mean, the possibilities are enormous. Imagine creating modified genomes to study specific diseases and then using that knowledge to develop totally new treatment approaches.
So we could see targeted therapies for genetic diseases, cancers, all sorts of complex illnesses that have a genetic component. This could really change healthcare as we know it.
Exactly. And it goes beyond treatment. Think about the possibilities in biodesign. We could engineer crops to be more resistant to disease or to climate change, or even create entirely new biomaterials with specific properties. The applications are just vast.
This opens up a whole world of possibilities, but it also raises some important questions about the challenges and ethical considerations that come with such powerful technology.
Oh, absolutely. There are definitely challenges. We're talking about incredibly complex systems, genomes with millions or even billions of base pairs, these really intricate interactions between genes. It's a huge challenge to synthesize them accurately.
It's like trying to recreate a whole rainforest ecosystem in a lab.
Right.
Just a massive undertaking. What are some of the other challenges you run into when working with systems this complex?
So one big hurdle is something called genome annotation. It's basically the process of identifying and labeling different parts of the genome.
Kind of like making a map of it.
Exactly. And even though we've come a long way, our understanding of the genome is still incomplete.
We don't have all the pieces of the puzzle yet.
There are still so many regions we don't fully understand what their function is. And that lack of knowledge can lead to unexpected problems when you're designing synthetic genomes.
So even with the best intentions, you could accidentally change or delete something important without even knowing it.
That's the risk.
It seems like even with amazing technology like Evo2, there's still a lot of uncertainty because these systems are so complex. Are there other things beyond just the biology that we need to be thinking about when we're developing and using this technology?
Oh, yeah, definitely. One challenge is what we call the black box problem. It comes up with some AI models, especially those that use deep learning. These models can be really complicated, and it can be hard to understand how they're arriving at their conclusions.
So it's like having a chef who can create a delicious meal, but they can't tell you exactly how they did it. You might enjoy the food, but you're left wondering about the process.
Exactly. And that lack of transparency can make it harder to validate the results and make sure the AI is really working on solid scientific principles.
That makes sense. So we've got this amazing AI that can design these genomes, but then there's the challenge of actually building them, right? What are some of the technical hurdles there?
That's another big area, yeah. Synthesizing and assembling DNA has gotten a lot easier, but it's still a very complex process. You have to make sure everything is accurate and reliable and safe at every step, especially when you're dealing with something as fundamental as DNA.
So we have the AI that can create the blueprint, and then the challenge of making that blueprint a reality in a safe and reliable way.
Yes.
It sounds like a real balancing act between pushing the limits of what's possible, but also being responsible and thinking about the ethical implications.
Absolutely. And figuring out how to navigate all of this requires a lot of careful thought and collaboration between scientists, ethicists, policymakers, and the public. We need to have open discussions and be transparent about what we're doing. That's really crucial as we move forward in this new and exciting field.
It really feels like we're at the very beginning of something huge in biotechnology with all this. So much potential. But we really need to think carefully about what it all means.
Yeah, EVO 2 is a perfect example of how AI is changing how we understand biology. It's opening up possibilities we used to only dream about.
Like science fiction stuff, right?
Right. And now we can not just analyze, but actually design and create genomes.
Incredible.
This could totally change how we approach disease, agriculture, even how we define life itself.
It's amazing what humans can do when we put our minds to it and keep searching for knowledge. But like with any powerful tool, we need to use this technology responsibly.
Yeah.
Because being able to manipulate the building blocks of life has huge ethical and societal implications.
Yeah.
And that responsibility isn't just on scientists. It's a conversation that needs to involve everyone, policymakers, ethicists, the public. We need to be transparent and have open discussions and set clear ethical guidelines as we move forward.
I agree.
It's going to be a fascinating journey. It'll challenge what we think is possible and make us ask some really big questions about the nature of life, the limits of technology, and our responsibilities as caretakers of this planet. Thanks for joining us for this deep dive into AI and genome creation. Until next time on Le Podcast de Philippe.